from MicrobeTV. This is Office Hours for Wednesday, February 11, 2026. I'm Vincent Racaniello, and welcome to your viral corner of the internet, where you can learn all about viruses and many other things as well, and appreciate the wonder that is science. Hello, everyone. How's everybody doing tonight? Welcome to Moderators. Les is here. Vanity Nutrition is here. Tom Steinberg is here. Thank you for coming and moderating. Thanks, everyone else, for coming. What we have on the agenda tonight, we have a little bit of news. Boy, the bad news about science just keeps coming. And we will have a mini lecture, maybe a quiz if we have some time, and a poem, of course, to wrap up the evening. Tell us where you're from. We'll shout you out. Yes, I was here on time tonight. Caught the 6:03 out of Newark. So I got out of lecture on time because when I'm finished, usually many students come up and ask questions, which is great. But tonight there were only 3. So I was able to get out and get the subway and get down in time to catch the 6:03. So let's see. Well, this individual, whose name I can't pronounce, is that Cyrillic? I just don't know. Is interested in memorizing virology and says, I could write down the same sentence 5 or more times. The problem is you will forget it if you don't use it. Like everything. I remember virology because I think about viruses every day. I do podcasts every week. I teach in the spring. I revise the textbook. So I remember it all. But, a lot of microbiology I forget because I don't use it. Microbiology meaning bacteria, fungi, etc. So you could remember it, but you'll forget it, unfortunately. But I think the key is to get familiar with the concepts, right? Remember the principles, and then you can apply that to everything. So also asks, how do you think would the contents of your great lectures be enough, or is it better to write down the essentials of each chapter? Well, the book is very comprehensive, right? You can't remember all that. I think if you could learn what's in the lecture, those are the essentials. Those are the essential principles. And I think you could always supplement that with the book if you wanted to. But I would take a lecture and watch it and take some notes and then go to the book and expand by reading the corresponding chapter to the lecture. It should be clear which chapter goes with which. Viruses Luna is here. I'm very sorry, Viruses Luna. Rima lost her dad. Sorry to hear that. But welcome back. Here's the Cyrillic person. Dear Teacher Racaniello, thank you very much for all your work. You're a true mentor, a gift to humanity. I have a question. I want to memorize very well the basics of virology. So the principles. I think you can learn them and then come to these or listen to the podcasts every week. And that will reinforce what you've learned because you'll hear things and you go, wait, I learned that. Wait, I learned that. If you really keep at it every week, then you'll remember it. We often have discussions here that will invoke what you've learned. So that's what I would suggest. Red Bobby from Idaho. Now Rita is in Idaho. I don't know, is Rita Red Bobby? Or do we have 2 people from Idaho, 44F and 7C? By the way, here in New Jersey, it's 0. Today was above freezing. It's 0 right now. It's going to go below 0. But it has not gone above 0 during the day for weeks, so this is a good sign. Another account where I have a different fake name, so I don't know if that's Rita or not. But anyway, hello, JJ from Southwest Florida, 17C. Thank you, JJ, for your support of science communication here at MicrobeTV. We really appreciate your support. Let's see, Linead. I just read a paper called Engineered Deletions of HIV Replicate Conditionally to Reduce Disease in Non-Human Primates. So I just took a look at it. So what they did is they made defective interfering particles of HIV. So defective interfering particles are viruses with pieces of the genome missing, but they still replicate, and they interfere with the replication of the regular wild-type normal virus, right? And so they treated animals with these and show that they prevent disease. The only problem is, there's a latent reservoir of HIV. Their DNA integrated into long-lived memory T cells, and this is not going to take care of that. Now, maybe if they show these persist for a long time, they establish their own reservoir. I don't think we're going to have a therapy where the therapeutic agent establishes its own reservoir or doesn't get rid of the provirus. I mean, it will prevent recrudescence, right? The reemergence of disease, which is currently carried out by antivirals, which have their issues, right? Now and then you get resistance and so forth, even with triple therapy. So we do need something else, but I just don't know if I worry that it will be combined with the wild-type virus, and then you're starting at the beginning again. And they say they didn't detect that, but just because you don't see it doesn't mean it's not going to happen. Anyway, those are my thoughts. Tom is in the Oregon Coast Range. Sunny, mild afternoon going to evening. John is in Minneapolis, where it's 0C. This is sad news. Dad went to the carbon cycle. Yeah, indeed. Hello, Visto from Sydney, 23C. Yeah, well, this is very interesting. So, on my screen, you see, moderators still have a wrench on the actual stream, but in the chat they have a shield now. I kind of like the wrench. The shield is nondescript. The wrench is cool looking. It's got some fine detail to it. JJ, listen to lecture 6. You mentioned that mistakes need to be made. Does the influenza virus make more mistakes than other viruses? Just to clarify, the mistakes are made during replication of RNA. Certainly, there are some mistakes made during translation, but those are less consequential because they're not inherited, right? They're not part of the genetics of it. They will not last for long because they get into the next particle, and then that particle will have the wild type gene or codon. So it's during the synthesis of RNA or DNA that mistakes are made. RNA viruses all make about the same amount of mistakes, about 1 in 1,000 to 10,000, an error an error for every 1,000 or 10,000 bases added or polymerized. And RNA viruses in general make more mistakes than DNA viruses because DNA viruses have an eraser. They can correct their mistakes. The RNA viruses don't have that. Mike and Lori is in Burien, Washington, where it's 9 degrees and clear. Hello, Patricia from Massachusetts. Nice to see you. Kang is in Chicago, where it's 1 degrees. There was sunshine today, and it is still clear. In Iowa, it is 5 degrees Celsius. All right. Kang asks, do bacteria have apoptosis? Yeah, they do. They have an analogous process. It has a lot of similarities, but it's not the same. It does have DNA fragmentation. It does have chromosome condensation. It has membrane changes. And it's triggered by stress. So it's like an altruistic suicide, right? That's why we entitled that episode, Would You Take One for the Team, or something like that. Because that paper was all about altruistic things. And then the measles vaccination was all about taking one for the team, right? Don't get me started. Anyway, yes, there are stress triggers like antibiotics would trigger or could trigger apoptosis. Penelope in Ecuador, welcome. Does DNA ever leave the nucleus? It can. It can, yes. During mitosis, the nuclear membrane breaks down, so it's technically out of the nucleus, and pieces of DNA may leak out. When viruses come in and those that go in the nucleus, they sometimes drop DNA on the way in as well. So yes, they do. Hello, Sandra. Welcome from Mexico. Barb tries. You see, now I got it since you corrected me. Durham, North Carolina, 13C. Elizabeth is in West Virginia. Maureen is in Northeast Ohio, where it's 28F. Lagodic is in H. Mark Martin, hello. Tacoma, Washington, 9.4C and sunny. Okay, that's Russian. Thank you very much. From viruses. Carol from Bakersfield, our favorite nephrologist, 17C. The viruses, Luna, will be in New York in May for a day. Okay. Maybe you can visit the incubator. IF Specialist is in Maine. Madawaska, Maine, 23F and snowing. Hello, Veronika. Dobry večer. Privyet, Moscow, snowy, -1C. Perfect winter weather. Oh, that name means firewolf. I was putting up before. That's a very cool name. Yeah. Mark writes, by the way, I actually went to one of Vincent's virology lectures. When I visited the incubator, it was fun to watch the students interact with him. And I also interacted with you. I love to interact with the students, yes. I always try and engage them and talk to them, not just be a shield at the front of the room. I walk around, I look at them, I ask them questions. I love it. As you know, Mark, you do too. Is from Queen Sirikit Park, which must be Thailand, I'm guessing. 27C at 8 a.m. That would be consistent. And that name translates to Agnenibolk. Wow, that's a tough one. Something like that. Lagodic is in Chicago, where it is above 0. Yeah. Dr. Oz begged people to get vaccinated only a year late. Yeah, what the hell? What the hell? It really takes you that long? Still needs to be fired. He doesn't know what he's doing. Mark Fuccio, hello. Central Coast of California. It's after 5 p.m. I've opened a 2018 Reserve Cabernet from Sarah's Vineyard. Sounds delicious. Sounds delicious. Good for you. And Leo is on the Mendocino coast where it's 56 and making a pot of macaroni and cheese. Lovely. Lovely. Hello, Betty from Washington Heights. 36F, home with bronchitis. Oh, George, sorry about that. Sorry. Barb remembers her typing exercise from the mid-20th century. Now is the time for all good men, people now, to come to the aid of their country. I took typing in the summer between 8th and ninth grade, right before going into. It was the best course I ever took, the most useful, because today I type, right, all the time. I don't need to look at I do proper typing. I forgot what that's called. Where you don't have to look at your hands and you don't do one finger at a time. No, I do proper typing. I used to get like 100 words per minute on a typewriter. Oh, man. And I often used IBM Selectrics. We had those at Columbia with the ball that has all the letters, and it goes around so fast, and it zips back and forth on the page, and the ball turns to get to the right letter, and and it's so different from a regular typewriter where every letter has its own key that pops up and smashes against the ribbon. Oh, it's such a hack. Listen, I remember I was pushing a Selectric on a cart at Columbia, and the cart got stuck, and it tilted, and the Selectric slammed into the floor and completely shattered. And that thing must have been $1,200 back in the 90s. Oh, gosh. It was a departmental typewriter, too. Nobody yelled at me. Nobody yelled at me. I like when you make a mistake and people don't yell at you. Now, if you do something intentionally, maybe you should get yelled at. I don't know. Hello, Frankie from Houston 22C. Hello, March Yvonne from the Philippines. I bet it's hot down there. Here's a little Russian conversation with, I guess, the person who was talking before. Peter. Dixon seemed to remember everything about everything. He was amazing. Miss him. Yeah, I do miss him too. It's a year now, roughly, since he died. Pete, we're 1 hour closer to you tonight in the Cape Verde Islands on the island of Sal. Days are about 23, 24C, but windy. 19C now. Here we go. Thank you very much for your kind answer. I'm interested only in one virus, but wanted to remember the essentials. We'll work on it. No, you have to be interested in all viruses. You can't be interested in just one, because you will not understand that one virus unless it's in the context of all viruses. Believe me, I've done this for 45 years. I've thought about viruses. You need to learn the main classes, the 7 classes, learn some representative viruses. Well, that's what I do in my course, so it's not a problem for you. Thank you, John, for your support of science communication. It is greatly appreciated. Thank you for enjoying the lectures. It's Rita. Okay, Red Bobby is Rita on a different device. You know, you have red instead of blue, and it gives you a different aura. MN is in D.C., where it's warming up. Sandra is in Mexico, where it's 22C. Call Me Ishmael for now is in Maryland, where it's 3C. Cheryl is in Santa Rosa, 53F, and light rainy. Okay, so Veronika's checking to see if it's someone she knows, and that someone is talking in Russian. They're talking back and forth in Russian. Okay, no problem. Aurelian, enjoying your channel from Vancouver. Recently read that biologically, every woman is a sexual mosaic. Well, no, not every woman. Some women are sexual mosaics, right? And there have been a couple of notable stories about this recently. One was a paternity case. The couple was getting divorced, and they did a biological test, a paternity test, and they said the baby wasn't hers. And she was pregnant and gave birth to another child, and they said it wasn't hers either, even though she gave birth to it. So then they looked into it, and it turned out she was a mosaic. Her germ cells were a different X chromosome from the rest of her cells. So it's really interesting. It's really interesting. And never anthropomorphized viruses, they hate that. That's funny. That's very funny. It's really an insider joke. Christina is in Hershey, PA. It's 30F. Jill is in Wilmington, where it's 38F. Downright balmy. It's a chat system in Telegram where science fans united in 2020 to discuss Sputnik, COVID, and vaccine. We sometimes Twitter there. Anyway, happy to be fellow Russians over here. Okay, cool. Is this person right here in Russia at the moment? Tell us. Give us a name that I can call you by, because I don't do Cyrillic. Pat is in Nova Scotia, where it's minus 1C, and where less is, it's 285 Kelvin. Ryan, hello, Ryan. How are you? Mary asks, obviously, if someone were to use an out-of-date COVID test, the test may not work, give a false negative. My question, would there be an increased chance of a false positive? I don't think so. I think that the failure of the reagents would give you a false negative, most likely. Mark is in Vegas, 18C. Robert, hello from New Jersey, not too far from you. I'm in New Jersey as well. Visto vaguely remembers the talk of using virus to lyse cancer cells. Visto, listen to my lectures. Come on. It's lecture 25. It's the last lecture in the course. Therapeutic viruses. And many of them have been tried to lyse cancer cells. VSV, measles virus, herpes virus, Rio virus, polio virus. All still, I mean, a few have been licensed. Herpes virus has been licensed in the U.S. to treat untreatable melanoma. And one has been licensed in China, an adenovirus, I believe. That's another one. Yeah, people are working on it. It's going to happen, but it's not there yet. Lecture 25, Therapeutic Viruses. GSD Bio, Jonathan, and Sunita, 16C, and Sagi. Okay, EHS Talent says, so to be clear, Daniel and the rest of us no longer trust T cells to protect, so we're supposed to get boosters semi-annually, correct? I just want clarity on what to do. So Daniel has always supported annual vaccinations. They don't call them boosters, just another vaccine that matches the circulating strain. I think it's more of an issue in people over 65, where any infection can cause a problem, whereas if you're younger, the T cells will help you out, even with divergent strains. So if I were 40, I wouldn't be getting, per Daniel's advice, I mean, everybody can get it, right? That's fine, because obviously even young people get sick. But I don't think it's as necessary as it is for people over 65. But, you know, kids do die of COVID. They should be vaccinated. Certainly they should get the primary series, right? A booster, annual booster is another thing. Again, I think 65 and older is the best use case for that. Carol got a picture of me and my granddaughter wearing our double helix hats while reading the double helix book together. Adorable. That's great. Carol purchased, or I don't want to say purchased because we're not allowed to sell things. We give things away for your donations. Okay, so she got hats, 2 hats, and that's just adorable. That's cool. I mean, you can send it to me. I won't share it. I'd love to see it. To my email. Ryan, good evening, Professor. I'm a PhD student in veterinary virology, and I would like to know, for the detection of viruses, a PCR viral culture in urine and fecal samples, should we add virus transport medium and store in, I don't see a follow-up. Yes, you should, if If you're going to bring the sample somewhere else, you should put it in virus transport medium for sure. Tractor Poodles in Bangkok, where it's 27C and sunny. Touch typing. Thank you, Les. I learned how to touch type. So I don't have to look at the keyboard. So they used to say when you had a piece of paper in the typewriter, don't look at what you're typing or the keyboard. So I'm like, what am I supposed to look at? Usually you were typing something, you were copying something. You could look at that, or just look around. If you're just writing, you don't have to look at any. Or sometimes you want to see what you're writing, right? So I don't think that's so bad. John, this week's TWIV talked about how phages are sensitive to the health of the bacteria because they need to know when to pull the eject mechanism. That's right. That's very good. I like that. Google AI remembers everything for us. It'd be lost without it. It's a bit like talking to Tom Riddle's diary, though. So, lately I've been talking to ChatGPT a lot. It's not Google AI, but, it's a similar idea. And I feel like I'm talking to someone. And so today I said, how do large language models work? And they say, is it basically you predict the next text, right? So if you ask it a question, it puts a string of words together and it makes predictions of what the next one would be by looking at this all of this information that it's stored. And I said, so really it's just about predicting the next word? Actually, it's a syllable. It's not even a full word. And it says, well, you're conceptually right, but you're missing all the details. It's like saying physics is about motion. It's more than that. And so then, it went on to explain to me all the other things that go into it. And then I said, well, when you make a mistake and I correct you, does that get fixed? And it said, no, it doesn't. It never gets fixed because people could game our database and screw it up. And so I felt like I was having a conversation with a colleague who was teaching me about large language models, right? And then, of course, at some point it says you have to pay now to get any more questions because I'm on the free model. But I think it's much better than it used to be. And I often use it to find out things like, how do I install a VPN on my home network? And he said, yeah, you can buy the vanilla stuff, that consumer VPN, but it's really better if you install it on a local computer. And I said, well, what about if I put it on a Raspberry Pi? And it goes, that's a great idea. It's always complimenting me. When I have a good question, it says, that's a great question or that's a great idea. But sadly, all the interaction is forgotten. I'm saying this because you say AI remembers everything. Yeah, you have to ask it. I mean, you do have a storage of your dialogue locally, but it's never, whatever you do with ChatGPT, they forget from the main database for reasons. But they really explain to me how they do this. It's very, very interesting. All right, so Will, hello, Cloudy, 7C, and Changsha. Oh, I didn't know this. Dr. Oz was found in the Epstein files today. Well, I can tell you that I'm not there because I never heard of the guy before he killed himself, frankly. And, this is disgusting. In every other country, if you're a pedophile, you're finished as a politician or as anything. And here we have, this Howard Lutz, whatever, the Yutz, he's really a Yutz. He lied about his association. He should be fired. And they're protecting him. Just like they're all surrounding the campfire, these idiots. They're going to go down. They really are. You cannot have, they say Trump's name is there 1 million times. All right, so Dr. Oz should be fired too. What is he doing associating with these guys? Even if you didn't do anything with young girls, you associated with this guy who was. Why? That's disgusting. Okay, so much morality from me. Martin, Vincent's cocktail from a recent ASM microbe. A rum old-fashioned. 2 ounces of dark rum, 1 teaspoon allspice dram, 1 teaspoon demerara syrup, 2 dashes Angostura bitters, 2 dashes orange bitters. Yeah, that was in Houston a couple of years ago. I asked the bartender to make a rum old-fashioned. It was really good. They had to climb up and get rum from way up, really top shelf. Patricia, most people had to take typing in junior high or high. It was an elective, so I didn't elect it, but I taught myself using basketball blocking dribbling goggles. Interesting. Worked well. Yeah, it was an elective, too. My mother told me to take it. She said, you will thank me, and she was right. It was really a great course. And the funniest thing, the teacher was Mr. Nasser. So Mr. Nasser was Egyptian, but he grew up in the South, so he had a Southern accent. It was so cool. He was the kindest, gentlest guy, though. He was really, really a great teacher. I'll never forget that Southern accent. Oz invited him to a holiday party. Why? Why would you have this crook, this disgusting, revolting human being? Frankly, revolting. Why would you invite him to a party? Maybe that's not enough to get you in trouble. But we'll see. There's a lot of stuff in there that, they let the Congress see it unredacted, but they can't copy it or anything. They just can take notes. Who knows what's going to come out? Up to Code Danny in Nova Scotia. -1, but it feels like -7. Snowing maybe all night. Nova Scotia. Cool. Cool. Yeah, Visto typewriters now sell for $1 in antique stores. The one's not broken by Vincenzo, that is. Yeah, I did break that one. Oh, my gosh. The Dirndl Girl. Hello from Longmont, Colorado, where it's 10 degrees C. Jonathan, you brought up the problem of detecting nucleic acids or proteins as an indication of infectious virus presence. Could one add non-canonical amino acids? I don't know, Jonathan. Please. So you're saying that these would be... I don't know how that would show you infectivity. Explain, please. Eliza, last week's office hours was great. Loved the mini lecture. -2 in Central Ontario. We'll have a mini lecture tonight. We're going to continue with capsid assembly modulators. Spamlet 26C in the kitchen this time. Hans, hi Vincent, they vaccinate wildlife against rabies, spreading baits with the vaccine over forests, etc. Wild animal eat the bait and get vaccinated. Why can't we vaccinate people via the oral route? That's a good question. Because we haven't tested it. And people, vaccinations approaches have to be tested. And there is currently an injectable vaccine. So I'm not sure there's any impetus to make an oral vaccine. But, the vector in the animals is vaccinia virus. So it's totally different, and you'd really have to go through testing to get it into humans. Cheryl just started reading science journalist John Cohen's new book, Planning Miracles, How to Prevent Future Pandemics. Are you familiar with this book? Is it worth finishing? I don't know. I didn't know he had written a book. Let me look into it. I mean... John is a science writer. He writes good things, but what is he? He's probably talked to a lot of people about preventing future pandemics. If that's something you're interested in, everybody has their own opinion. I'm not sure how compelling it could be, but let me look into it. I'll find out who's read it. Oh, Jonathan continues, adding AHA to mucosal tissue, that is, and checking antigen by both AHA incorporation and with the appropriate antibody from the LFA. It sounds interesting, but I've never heard of that before, so I can't even comment on it. Huh. So how do you add it to me? What are you going to do, spray it or swab it into the nose and then pull it out? John writes, what does it mean to crystallize a virus? I have a hard time visualizing the process, and why is it more difficult for some viruses and easier for others? Well, just think of salt. Those are crystals, right? Put a few grains of salt in your hand, and they're pretty regular. Or take a big chunk of quartz, right? Nice big chunks of quartz that you can get in geology stores. That's a crystal. So, I mean, for the virus, the crystals are really small because you can't make enough virus to make a big crystal like that. But it's essentially what it is. All the viruses glom together in a lattice that basically makes this regular, ideally well, Frankie actually, maybe Frankie has done this and he can give more insight, but which is a regular structure. And when you bombard it with X-rays, they will bounce around all the atoms and give you reflections that relate to the structure. Retired researchers in Wales, UK, 1:21 a.m., cold and dark. Wow. Wow, this is very brave of you to be here at this hour. Kip and Laura, thank you for your contribution to science communication. We marvel at your long-term memory recall. You and I are the same age, and I hope I can keep up with you as we move forward through our mature years. I think the key to long-term memory is just to use stuff over and over. Although I can come up with names sometimes of things that happened years ago, and it actually surprises me. I don't know why. I do use my brain a lot. Maybe that helps. I always have, right? I always had to think to do everything I've done from graduate school and postdoc and being a research professor and then podcasting. So I think that helps a lot too. And when I can't remember something, I just take a break and I try and do other things, and it'll pop into my head at some point. Microbio in Burlington, it's 0C. Welcome. Those golf ball typewriters, those are the IBM Selectrics. It looked like a golf ball, and when you typed, the ball would spin around to the right letter. Had Russian spyware in, I recall. They fitted out the U.S. Embassy with them. Oh, I can imagine you could do that, and then you could read what's being typed, right? Women aren't mosaics. They are mysteries wrapped up in enigmas. That sounds like a saying I've heard before. Trion are transfected with mRNA that encode and induce cell to make non-self-antigenic proteins targeted by T lymphocytes. They can be. It depends if they're T-cell epitopes, right? Not every part of a protein is a T-cell epitope. It has to be a certain length and fit in the T-cell receptor. And so there are algorithms you can use, right, to screen an amino acid sequence and identify T-cell epitopes, and then you can check them out. But, not every amino acid in a protein is a T-cell epitope. Alexi, welcome from Toronto. It is -2, feels like -9. Your programs are like a beam of light in the kingdom of darkness. Thank you so much. I really appreciate that. I love that people love what we do. I mean, I have fun doing it, but obviously it's the most fun when people learn from what we do. Pete popped over $50 a week back on your contribute page. Hope you get most of it. Thank you, Pete. I really appreciate your support. Razor. I tested flu 2 weeks ago, still feeling under the weather. Avoid flu if you can. Yeah, it's a minimum 2-week illness. Yeah, feel crappy for a couple of weeks. Yeah. Aurelian writes, one of the 2 X chromosomes in each somatic cell is randomly silenced. That's what I meant by mosaic. That's correct. Yes. One of them is silenced. Sandra, thank you for your contribution to science communication. Really appreciate your help. MN writes, my silverware is called Sasaki Double Helix. I've had them for decades. Oh my, I have to look this up. Sasaki Double Helix flatware. Oh my gosh. Look, here it is. Okay, I got to show this to you. It's pretty cool. Here we go. There it is. See, it's got this little twisty action going on there. So this is dinner forks, set of 4. Very interesting tines there. I like that. $100 for 4. It's a lot of money. But if you like them, I saw some others here too. Sasaki Double Helix flatware. Here's a fork for $59. See, there's the twist in the neck. That's where the double helix come. I guess these are, that's replacements limited. These are not made anymore. That's why they are expensive. And here's a spoon with the same twisty there. That's very cool. Cool, cool, cool. Thank you for pointing that out. I like that. All right. Let's go back to me. According to Les, the Russians built the U.S. Embassy. Even the U.S. seal had microphones embedded. Good night, Mark. Have a good evening. Have a good dinner. Tryon writes, did the intradermal way of injection mitigate the risk of blood spread of liposomes with mRNA? I'm not sure that it was done very much, and nobody did a study to look at that, right? So I think the intramuscular was the way to go. But sometimes, as a retired researcher says, intramuscular injection mitigates LNPs entering the bloodstream. But, one of the solutions was to put the needle in. It's a deep injection. Pull back and make sure there's no blood so you're not in a blood vessel. But nobody really did that. Nobody did it. What if I put it on a Raspberry Pi? Did I hear that right? Yes, you did. A Raspberry Pi is a little computer, right? You must have heard of that. They're very cheap. They're like, the board is about this big, and you can plug power into it, Ethernet, a monitor, a keyboard, and a mouse, and it's a full-fledged computer. And it can run headless, which means without a monitor. So you can run, and its power requirements are like 5 watts a day, as compared to a computer, which you leave running and run your VPN, which would be 60 watts a day. So it's more efficient. So anyway, I was asking ChatGPT about that, and so there is an example where it's useful. And so here I go. How do I do this on my phone? And it said, okay, go into the menu and look under this and this. And I said, it's not there. What you say is not there. And it goes, oh, you're right. They've updated the interface software. Sorry. But that is never going to get incorporated. So when another person asks the same thing, they will get the same mistake. But what ChatGPT did tell me is that they have people watching answers anonymized. And if they see a lot of mistakes to a particular question, they will fix it. He said, and Chat said, it's like a textbook. It has a mistake in it. People notice it, but it doesn't get corrected in the textbook until the next edition. And so that's, you send your corrections into the publisher, and eventually they come into the next edition. So that's the same thing with ChatGPT. Yeah. Did scars occur after the skin patch use with mRNA that induces antigen? No, I don't believe so. I don't believe. I don't believe that skin scars occurred. Howard Lutnick, thank you. Yutnick, is he Yuts? Yeah, the Pi Mini computer. That's absolutely right. Should have seen the Bondi hearings today. So I tried to. I mean, she's just offensive, right? And I couldn't read it in the Times because I don't have a subscription. But they said she would not apologize for anything to the victims of Epstein. Yeah, here's Raspberry Pi to find out more about that. Vincent Spamlet writes, the Google search AI does all you mentioned and will teach you to do anything you ask for free. I've hit no limit yet. It likes talking and knows everybody. Turing plus plus plus. So I tuned ChatGPT to the level of sophistication that I wanted, right? So like a scientist level sophistication. I don't want any silly things, and I like it a lot. Well, I'll try the Google AI. What is that, Gemini? The Google search, not just search, but a real thing where you go to the page and you type in queries, right? Jill writes, if AI gives you bad information, how would you know? Well, so that's why you have to be careful what you ask it, right? So I ask it, how do I find out the subnet address of my network, right? And so it says, go into your iPhone and look at this setting. And there is no such setting. And I told it that, and it says, oh, they updated the software, but it didn't give me a solution. It said, try this instead, another thing entirely. So in some cases, you could pick up the mistake yourself. If I ask it about viruses, I can tell when it's wrong, right? But that's because I know that stuff. So I don't ask it other things. Although today I wanted to know, how do large language models work? And I don't understand half of it, but let me show you. I think I can pull it up here. It's really an interesting exchange. All right, so I'm going to show you this little exchange. And let me take Jill off here, and we'll go back to the screen share. Okay, so this is, I asked it, how do LLMs work? Large language models are autoregressive neural networks trained to model the probability distribution of token sequences, and this is the formula for it. They learn to predict the next token given all previous tokens, and then they break it down, right? So raw text is segmented into subwords, like unbelievable is unbelievable, and they map it to an ID and so forth. And then, I don't understand, modern LLMs are built from stacked transformer decoder blocks. I don't know what that means. So it could be right or wrong. And that, it keeps going on. They're very verbose and repetitive, but I think that's okay. Why they seem intelligent? They do, and I don't understand how that can be for something that's just figuring probability of the next syllable. Due to scale is a big thing. Deep hierarchical representations and attention enabling structured reasoning pattern. They learn grammar, world knowledge, coding syntax, scientific discourse, and stylistic patterns. However, they don't have goals or belief like viruses. They don't understand in a biological or phenomenological sense. They approximate distributions over language. And they also get supervised fine-tuning, right? There are instruction data sets and reinforcement learning from human feedback, etc. Limitations, hallucination, confidently wrong outputs. So, Jill, that's what you're talking about. Context window limits, no persistent memory, no grounded perception, no intrinsic truth-seeking. They are fundamentally probabilistic sequence models. Okay, so that's great. So I write here, are you saying that ChatGPT is all about the probability of the next word? Short answer, at the training objective level, yes. At the functional level, no. That's an oversimplification. Let's unpack that carefully. I mean, it's like talking to me. It's a little off-putting, right? Because this is not a person. And so it goes through a lot of this to explain why it's not just token. And so I say, so it says, and I said, I don't understand how ChatGPT answers a question by next token likelihood. What am I missing? And then it says, good, this is the right confusion to have. So it's like being a teacher, it's telling me I asked a good question, which is what we all do, right? The missing piece is this. The model is not predicting the next word. It is predicting the next word of a completion that follows your question. That distinction matters. And so it gives something, it goes through this, and that's very cool. So let me tell you the next question I asked. So does ChatGPT learn from every question it's asked? No. ChatGPT does not update its weights from each individual question in real time. And then it explains why it doesn't do that, okay? And then I say, sometimes ChatGPT gives the wrong answer. How is that taken into account? Good question. There are two distinct levels, during the conversation and across model development cycles. Okay, so that's fine. And it explains why this is so basically this, where is it? There was a good analogy. And wrong answers still occur. Okay. Humans are warned from a young age to learn from their mistakes. For ChatGPT not to do so seems counterintuitive. It feels counterintuitive because we're importing a biological learning model into a static trained system. And it goes on. Okay, so I find this a really unsettling conversation. It sounds like a human being is talking to me. And for the most part, it makes sense. And so that's what you just have to be careful what you're asking about, and you have to tailor your questions in the right way. And I'm still learning, right? So I'm having a conversation to understand how it works. Well, all the interaction is forgotten. I'm not sure that's correct. It just told me that, right? But for you, it's stored in your browser cache. That's the thing. It's not stored anywhere else, just in your browser cache. We're waiting here in the UK to find out which former prime minister was involved with Epstein. Yeah. Dorothy Charging Hawk 26F in Duluth, February thawed tomorrow above freezing. Tom Hanks owns over 300 manual typewriters. Wow. They are fun. I don't have one anymore. It's unfortunate because I used to like them a lot. Okay. We shouldn't use it as a search engine or consultant. It will hallucinate at some point, and you can't be sure when. It's also horrible for the planet. Yeah, well, that's the other thing. It's very bad for the planet. But I find it interesting to learn from it the way I was just doing. All right, let me take a break from questions here, and I want to give you some news from this week, which is just horrible. Okay. So this is an article from Wired. Gosh, let me put up the screen here. I don't want to see you. What's your face? Okay. RFK Jr., this is by David Gilbert, picks for key autism panel include advocates for bizarre theories. Oh, my gosh. So this is a different panel now. He has this autism committee. It's the interagency, let me make it bigger, the Interagency Autism Coordinating Committee, a group that recommends what type of autism research the government should fund and provides guidance on the services the autism community requires. This group is typically composed of experts in the area of autism research, along with policy experts and autistic people advocating for their own community. So he got rid of all the previous members, and he says he's appointed the most qualified experts, leaders with decades of experience studying, researching, and treating autism. But health experts and autism advocates strongly disagree. And a review of the new members suggests he appointed members of the anti-vax community who claim that vaccines cause autism, despite there being no evidence to prove such a claim. Oh, my God. So one of the appointees was Daniel Rossignol, a doctor who was sued for fraud after prescribing a 7-year-old autistic child a debunked and dangerous treatment. Another appointee, Toby Roberts, has claimed that no thinking person vaccinates and that vaccine makers are poisoning children. He is a fellow at this institute and called vaccines one of the greatest crimes in human history. Other appointees are no different. They list some of these other appointees here. And I just want to say a few more things on this. Just a few years ago, this may have sounded like the all-star lineup of a conspiracy committee. Today, these appointments appear routine and just the latest example of how Kennedy has sought to remake America's public health administration. And this is a quote by Gavin Yami, professor of global health and public policy at Duke. Once again, Kennedy proves that he is one of the world's most extreme and dangerous conspiracy theorists who love stacking his committees with anti-science, anti-public health kooks. And then we have a quote from a lady here. Where is it? So an autism activist, Fiona O'Leary, we've fought so hard to protect our community, but they want to experiment on us. She has spent years fighting the spread of baseless treatments for autism. We are guinea pigs. When you are autistic like me and a mother to autistic children, I see this as a way to end us. That's what it is. I mean, can you believe this? Can you believe this stuff? All the whack jobs are in power. Okay, so that's one. Then there's another one that you may have heard. This is from The Guardian. What is this? I can't believe. FDA declines to review Moderna application for new flu vaccine. So Moderna submitted data to request a review for licensure of a new flu shot based on mRNA, right? And FDA said, we're not going to review it. What? That's unprecedented. You always review submissions. First of all, that's how the FDA gets its operating budget, because the companies pay for the review. And secondly, why would you ahead of time not review something? It doesn't make any sense. So this is likely to discourage industry from investing in future vaccines, right? Why? Just because Marty Makary and Vinay Prasad don't like mRNA vaccines, they don't want any more mRNA vaccines, so they will not review the evidence from Moderna's clinical trials. They say because the trials compared the Moderna shot to existing flu shots rather than shots for high-risk individuals. That's their reasoning for not considering it, because they don't like the comparison. But that is actually the way it should be done. You should compare the mRNA vaccines with existing ones. The FDA doesn't consider the Moderna trial to be adequate and well-controlled. You know, they don't know what they're doing. Prasad has no idea what he's talking about. So people say, as this person says, for under 65, the high dose is not standard of care, so the argument is false. So this is really bad because other countries are going to review these vaccines. And the FDA, the application is already under review in the EU, Canada, and Australia. So this is horrible, folks. They are, they're ideologues who have views of vaccines, and they are going to impose them on all of us, even though most of us did not vote for them. That's not the way I would like it. I want this vaccine reviewed, right? That's the way it should be. Let's go back to some comments, and then we'll do a mini lecture. Frankie, I have tried X-ray crystallography. Solved a previously solved structure too. Unfortunately, I wasn't able to get any novel structures. So this is the thing: is the Attorney General of the U.S.'s job to impede justice? No, the Attorney General is supposed to work independently of the president, but it's all screwed up in this administration because he picked someone who would just be loyal to him. And it's disgusting. I want her to be in jail one day with the rest of them. But she in particular has just lied left and right and done things that are completely unconstitutional. Retired researcher writes, in the UK, the Reform UK Party is likely to have Dr. Asim Malhotra in a similar role to RFK Jr. in the US. He is notorious for his comments on COVID vaccines. Patricia writes, the job of the Attorney General is to scream at people who do not recognize the president's superiority to every living thing on earth. Geez, did you not watch? Tongue in cheek, of course. The worst part of crystals is it's random. No one knows how to exactly things crystallize, so lots of screening is needed, and it takes a lot of protein each round. Right. Am I sleeping any better? Marginally, but still not good enough. There you go. Russia is a riddle wrapped in a mystery inside an enigma. Thank you, Vanity. Thank you very much. Even cryo is hard, and my cryo sample prep took years to get to work and involved quite some luck, too. $59 for a fork. Fork off. When you see a fork in the road, take it, as Yogi Berra said. Yeah, double helix silverware. I will grab it if I find it in a thrift store. Tom writes, I thought you were meant Raspberry Pi and it was a joke, revealing that I'm out. Out of it are e-devices. No problem. It's fine. Bondi was a troll. She acted like a sassy 12-year-old. Wonderful. Google's Gemini is very impressive. Okay. I will check it out. Microsoft Copilot is a bit sassy in her replies. Ryan Lamraoui. I'm sorry. I'm from Algeria, and it's currently 2:47. Well, welcome, and thank you for coming. I hope you enjoy it. On a lighthearted note, for the day when the only Epsteins I knew were Brian Epstein of the Beatles and the Epstein-Barr virus. Indeed. Indeed. Well, so Red Bobby says she asked chat how to germinate orchid seeds, and you got the wrong answer. You got peas. Haven't trusted it since, but I don't know. You shouldn't have to trust it, right? Don't give it such mission critical things, right? But I do think it's getting better. The AI chat of my days in nursing school was Wikipedia. Our A&P prof said, don't use it to study. I thought that was great advice, and I feel similarly about ChatGPT. Yes, don't use it to study. Don't use it to write your papers, but there's some situations when it's useful. What else have I asked it? I also use it to remove photo backgrounds. So when I make images for my video thumbnails, particularly of Daniel, I take out the books behind him. And ChatGPT does a really good job removing backgrounds of photos. And you can tell if that's done right, right? I wonder what ChatGPT would say if you asked it why RFK Jr. doesn't believe in the science of vaccine safety. Well, let's do this experiment, okay? Let's do it. Why doesn't RFK Jr. believe in the science of vaccine safety? Why? Let me put this up so you can see it. Why doesn't RFK Jr. believe in the science of vaccine safety? Okay. Oh, it's taken a while. RFK has publicly stated he believes vaccines are not adequately tested for safety and that regulatory agencies are overly influenced by pharmaceutical companies. And then the scientific consensus, why the disagreement persists. This is not a dispute within mainstream immunology about whether vaccines broadly work or are safe. It is a dispute between the overwhelming consensus of biomedical research institutions and a smaller group of critics who argue that safety oversight is inadequate. And then it offers to help me do other things, which I don't want to do right now. So there you go. I think it's an accurate depiction of what's going on. Okay. Hello, Questella. Welcome. It doesn't matter. It's just an hour in. We're going to do a mini lecture soon. And where does Tom Hanks keep all the typewriters? Well, he probably has a lot of space, right? So Spamlet, here's a good use case. I started out using Google AI to remind me of the word perpresture, and it taught me how to fix my PC downloading the Zorin OS with my phone. OTG lid to make a live boot drive. That's the kind of thing it's pretty good at, yeah. I used an IBM typewriter, John writes, briefly before a Mac SE with ImageWriter, too. I can remember the constant electric hum when it switched on, and the whiteout tape was mesmerizing to me as a kid. Yeah, automatic whiteout cake. Yeah, here's the link for that Wired story that I just put up. Elizabeth writes, RFK's obsession with autism and autistic people is seriously pathological and very disturbing. He is up to something beyond his anti-vax BS. So the Google AI is just at the left end of the search box choices. It's not a separate app. Just type a question and it will answer before you finish typing. Where is this? So you just go to the google.com. Okay, so you go to AI mode. So let's see, who's Vincent Racaniello? Let's see if it gets this right. Okay, it tells me who I am. It says I made the first infectious clone. I identified the polio receptor. I made a mouse model. I wrote the textbook. Science communication. I have pioneered the use of digital media for science communication. Founded MicrobeTV, my professional background. All of this you can get from the internet. So, okay, I will try Google. I'll see if it can remove the background, although I'm still using the free ChatGPT. It's just limited to how much you can ask it questions, right? Do you think the future Moderna mRNA vaccines could be approved and available outside the U.S.? Yeah. In fact, they are applying to Australia, EU, and another country I read. So, yes, yeah, Europe and the UK, for sure. It will be available. So you could always go and get your vaccine there. But oh man, I hope the rest of the world will be able to pick up the slack in research. Listen, in November we have a chance to reverse this. Everybody needs to vote against these guys. All of the local Republicans need to go. I'm sorry. Because I'm not trying to influence you politically, but it's the only way to reverse this situation because they're all standing around and protecting everyone else. Yeah, the FDA also said 10 children died because of the COVID shot. No, it's not right. There's no proof that 10 children died whatsoever. Just BS. Great podcast on brain docs YouTube channel discussing the cause, detection, and treatment of autism, over 250 genes. Yes, it's a genetic disease. There may be environmental components, but there's a huge genetic component. Yeah. Mary writes, as an uninsured poor person, I use the health department for vaccines. I went to get my shingles vaccine. The receptionist said, unfortunately, we are out of our $21 shingles vaccine because this administration is not going to help people, poor people, get their health care. There's no question. Thank you, Questella, for your support of science communication. Really appreciate it. Oh, continuing, Mary, the government cut our subsidy. They're now $250. Yeah, the government cut it. Bastards. They should be supporting people and people's health. That's the role of government. Unbelievable. Yes, you see books in Daniel's videos, but I make a thumbnail for YouTube, right? I take the books out. I like to have a black background with just Daniel sitting there. Just go look at the YouTube channel, you'll see. And the books are removed in the thumbnail, not in the video. They can stay in the video, that's fine. But just for the thumbnail, I take them out. Why take out the books? Because it's distracting. Okay, it's distracting. A friend, Les writes, a friend got me an Anthropic Claude Pro subscription. It is spectacular and programming. I had it write a program to create timestamps. So is this for programming only, or is it another chat GPT? Hello, California, Norma. Welcome to office hours. Yes. No, chat GPT is not the same as Google AI. Different companies, right? ChatGPT does a great job of repairing old photos and colorizing black and white images. Okay, let me do a mini lecture now before time gets away from us. Okay, because then we're going to do some other things. Mini lecture tonight. So, Tom, you asked for capsid assembly modulators, right? So this is part 2. Last week we did lenacapavir because the target is the capsid. And so this week we are going to do the hepatitis B virus capsid assembly modulators. So these are not yet licensed, but they're in clinical trials, as you will see. Okay, let's go to the videotape here. Capsid assembly modulators. So these are small molecule antivirals. They target the core protein of hepatitis B virus, and they disrupt nucleocapsid assembly. I will show you pictures that make this clear. They're among the most advanced non-polymerase HBV drug classes in clinical development. So there are polymerase inhibitors that are licensed for treatment of hepatitis B virus infection, but they do not get rid of the viral DNA, which is in the nucleus of cells. All they do is prevent the production of new infectious particles, but you have to take them forever. So people are looking for new approaches. So here's the reproduction cycle of hepatitis B virus. So here we have a cell with a nucleus, and the virus is binding to the surface. It's an envelope virus. And so the membrane of the virus fuses with that of the cell, and the core, which is the nucleocapsid, then docks onto the nucleus. So the nucleocapsid has double-stranded DNA in it. The double-stranded DNA, or partially double-stranded DNA, goes in the nucleus. It's repaired and then serves as a substrate for the production of mRNAs, which, of course, go out in the cytoplasm. They make proteins, and new capsids are assembled. And then these capsids that contain the viral DNA they acquire a membrane by budding into the ER, and then they're transported to the surface and released as the mature virus particle. So there's no genome integration with hepatitis B virus like there is with retroviruses. So these viruses remain episomal here. They're covalently closed circular DNA separate from the host chromosome. The other interesting thing about hepatitis B virus is that initially this full-length mRNA derived from transcription of the DNA in the nucleus, this is called pre-genome RNA. This is packaged into the capsid. And then, along with a molecule of reverse transcriptase. And then, before the virus even leaves the cell, it reverse transcribes the RNA and makes a DNA copy, a partial DNA copy, because it runs out of triphosphates. And so the virus particle that's released has DNA in it, not RNA, even though RNA was initially packaged. All right, so this is the capsid, this structure here in number 11, for example. That's the capsid of the virus, and that the capsid protein is the target of these small molecule capsid assembly modulators. So let's take a closer look at the capsid. So this is a envelope virus with glycoproteins in the envelope, and within the envelope is the capsid. And the capsid contains the viral double-stranded DNA. All right. So let's take a closer look. Here we have the structure of the capsid protein. So the capsid, which is inside the membrane, these are icosahedral structures. They're T equals 4, which means 4 protein subunits per structural unit, and they're composed of 120 dimers of the capsid protein. So here's the core protein, where the capsid protein is 183 amino acids in length. And so 2 of those form a dimer, and the dimer is shown here, and the dimer is repeated 120 times to make the icosahedral core. And you can see the different axes of symmetry. Here's a 5-fold axis, here's a 6-fold axis, and so forth, which you expect in a particle greater than T equals 1. So each monomer has a number of alpha helices, as you can see, that mediate the intra and interdimer interfaces, right? So this would be intradimer within the dimer, and interdimer would be between neighboring dimers in the capsid. Now, all of the capsid assembly modifiers for Hep B that have been characterized to date, they disrupt capsid protein assembly. They bind to a conserved hydrophobic pocket. Let me move this over so you can get me out of the way. They conserve a hydrophobic pocket at the dimer-dimer interface. So again, here's our icosahedral capsid with 120 dimers of the capsid protein. Here's a close-up, and here is an even closer view. So here are the dimers here in various colors, and we're looking close at the binding of one of these drugs in a hydrophobic pocket, and this is the interface between dimers here and here. All right, so that is how these drugs are binding to the capsid protein. Now, they were originally discovered by phenotypic screening. In other words, people simply look at collections of small molecules that would inhibit hepatitis B virus infectivity and then assembly. And initially, HAPs, heteroaryl dihydropyrimidines, were identified as drugs that suppressed replication but did not inhibit polymerase activity. So that was the key. They wanted inhibitors of virus replication that did not target the polymerase, because we already have lots of polymerase inhibitors. We want to find something new. And so they discovered the first capsid assembly modifiers with this kind of a screen. And then structural studies by these individuals showed that these HAPs, and again, the HAP is the chemical, they bind a hydrophobic pocket at the dimer-dimer interfaces I've just shown you. They accelerate assembly uncontrollably. So they accelerate assembly, and they make aberrant capsids and tubes and aggregates. So they make fracocta capsids, okay? They're all messed up because they're assembling too quickly. So these are now known as class 1 capsid assembly modifiers, these ones that accelerate capsid assembly and make aberrant capsids. But they actually have not worked out very well. So people then kept going back and looking for more CAMs, and the second generation CAMs were discovered. And these do not make fracta capsids. I mean, all messed up. They make ordered but empty capsids. So the capsids are normal as far as we can tell, but they don't have DNA in them. And these are chemically diverse with different scaffolds like phenylpropanamides, sulfamol, benzamides, and morphothiazidines. Different structural moieties comprise these new ones. So these are type 2. So again, these compounds promote the formation of morphologically normal capsids, but they're empty. There is no pre-genome RNA in them, and there's no reverse transcriptase. And the reason is that assembly occurs too fast and too early for RNA encapsidation. So the capsid proteins are present. They immediately assemble in the presence of the drug before the RNA can get in. All right, so these are now class 2 CAMs, and they're currently dominating clinical development. And they really have led to this key insight. You don't need malformed capsids to stop hepatitis B virus replication. You only need mistimed ones. In other words, you assemble too quickly and the genome can't get in. The capsids are normal, but without a genome, of course, they're not going to be infectious. So you're inhibiting viral infectivity, the production of infectious virus. So here's the summary. CAMs, class 1s, are misassembly inducers. They drive aberrant aggregates. They're malformed, unstable. They block encapsidation, and these are the HAP chemicals in those. And the class 2 are empty capsid accelerators. They promote rapid assembly, but they're empty. They lack pre-genome RNA and polymerase. They're ordered, but non-productive. And most modern clinical candidates are now class 2 CAMs. So this has been under development for a long time. So here, if you're a chemist and or interested in chemistry, these are what they look. Some of them look like, not all of them. So in the left column are the class 1s, and on the left here is the clinical trial phase that they have been through. So the core skeletons are blue, and these are nonpolar aromatic chains which go in the hydrophobic pocket. They're colored purple. So the fluorine and the bromide and the chlorine, those are highly hydrophobic, and they go into the hydrophobic pocket of the capsid protein. And on the right are the class 2s. You can see various names, all of them, again, having hydrophobic aromatic substituents that fit into the hydrophobic pocket. And so, and again, these are in phase 1 and phase 2 trials. So it's another summary. Again, the clinical investigation of these compounds began in the mid-2010s, still going on now. You can see they're mostly class 2 compounds. There's one, GSL4 is a class 1-2 compound being tested in China, but all the others are being tested by other companies, Janssen, Gilead, Novartis, Roche, and et cetera, et cetera. And many of these are in phase 2. This one has been discontinued, and this one is in a preclinical, early clinical trial. And so in the clinic, what do these drugs do? You get rapid declines in hepatitis B virus DNA, often 2 to 3 logs decline, and they work well with nucleoside analogs, which inhibit the polymerase. When used alone, the CAMs have a minimal effect on hepatitis surface antigen production. So they're not used as monotherapy. And if you do use them as monotherapy, you will get resistance mutations, which make them useless. And alone, they don't this statement is very interesting. From all the human trials, we conclude that alone, these capsid assembly modulators, they don't eliminate existing Hep B DNA. And that's why they have a limited impact on production of hepatitis B surface antigen. So they're used as in combination therapy. They're not going to be used on their own. But this is a paper we did on 1289, cell host and microbe paper, functional immune responses induced by a capsid assembly modulator in chronic Hep B virus infected humanized mice. So this is in mice, and actually the CAM did achieve elimination of Hep B DNA in about half of the mice. So here they have a humanized immune system and liver mouse model. So the liver and the immune system are from humans, transplanted in. They infect them with hepatitis B virus. And then they treat them with a capsid assembly modulator, GLP-26. Let's see which one this is, if it's on one of these. I don't think it is. Yeah, here it is. It's a class 2 at the lower right there. This is a class 2 CAM. And then you look at the outcome. Half of the mice, so again, this is Hep B DNA on the y-axis with time post-infection. So you initially infect the mice, Hep B replicates, that's the red line. Then you treat with the GLP-26, the DNA levels go way down, and you treat, and then when you withdraw the treatment, you have viral rebound. The DNA levels rise. And along with this, they notice they have a weak B or T cell response to the virus. So that's half the mice. The other half of the mice, after you withdraw GLP-26, viral DNA remains undetectable. And here you have very good B and T cell responses against the virus and seroconversion. So 50% of the mice do this. And we don't know why 50% do it or why 50% have rebound. But this is pretty exciting. And this deserves some investigation because it's just GLP, the CAM on its own, right? Whereas in humans, the CAMs alone never do this. So we have to figure out what's going on. I think that's very exciting. And so the goal, of course, is to achieve a functional cure of hepatitis B virus, right? And so to do this, we combine CAMs with nucleoside analogs, and then these nucleoside analogs suppress DNA replication. The CAMs prevent nuclear capsid formation. And sometimes you can pair these two with siRNA or antisense oligonucleotides to further inhibit the immune response, to lower the antigen load. And you can also add immune stimulants or immunotherapeutics to expand viral immunity. And these include therapeutic vaccines, checkpoint inhibitors, and toll-like receptor agonists. So people are trying many different things. And so this is very interesting because this is obviously extremely optimistic for Hep B, but it has effects beyond Hep B. It validated the capsid as a druggable system. Now, there were capsid inhibitors of picornaviruses years before, but they were not clinically useful. So this is really the first one to validate the capsid as a druggable. An allosteric system means you put a drug in and it affects, the assembly or some aspect of assembly of the particle. And these have a link to HIV and capsid inhibitors, right? Lenacapavir, even though they're distinct. Lenacapavir acts at multiple stages in different ways, but they are capsid inhibitors. And so assembly kinetics is now a therapeutic vulnerability that can be exploited for other viruses as well. So here's a typical treatment timeline to achieve a functional cure. So phase 1B, you have your CAM2 plus your standard nucleic acid analog. You look for tolerability, pharmacokinetics, viral load suppression. Then phase 2 would be CAM and your NA plus maybe siRNA or antisense oligonucleotide plus or minus immune modulators. You look for viral protein and DNA decline. And then phase 2 and 3, you optimize and eventually look for the if you get functional cure, if you confirm the efficacy versus your standard of care, you can look at the durability of the endpoint. So lots more to do here. We're talking about many more years of research, but very promising for, we have 350 million people globally infected with hepatitis B virus, and there's no way to get them free of DNA. So this could be one of the ways. So pretty exciting, I think. So thank you, Tom, for suggesting that I do capsid assembly modulators. So let's see what we have here. I'm going to give you a quiz, a brief quiz, okay? Because last week we did 3 questions, and you did really well. So we're going to do a couple more questions today to see how your virology is going, your virology knowledge. So let me just pull up the quiz. So last time we did what is a virus. So today we're going to do the infectious cycle. Okay, so now let's check this out. So remember, you go to I have to show you how to do this. Hold on. Go to Socrative.com and where is that? Log in as a student. I'm going to get you the information. Hang on. Go to Socrative.com, b.socrative.com, log in, student, and the room number is virus. Okay. Okay, and bsocrative.com, and the room number is virus. Thank you, Vanity. And then the question is, a blank and blank cell is the only cell that can take up a virus particle and replicate it. So fill in the blanks. And don't do it in the chat, okay? Because I want you to do it at Socrative so that we can have everybody answer and not have any issues with lagging. So naive and resistant, primary and permissive, susceptible and permissive, susceptible and naive, continuous and immortal. Which 2 words are the only cell that can take up a virus and replicate it? Don't worry about it. Where do you put the room number? Well, you go to log in and you say, bsocrative.com, login/student, okay? And it will ask for a room name. So you have to go b.socrative.com/login/student, and then it will ask you for a room name, and that's virus. So bsocrative.com/login/student, and John says, big and tall. Yes. So if you see here, we have 28 people who have got in, and 24 of you have answered. So I'll give you a little more time so that you can get in here. Cantankerous and immoral. Sounds like the administration. All right, let's see how we did. 25 people. Susceptible and permissive. 92% of you got that. That's great. Yes, those are the only. Susceptible means you have a receptor, and permissive means the cell is internally able to support viral reproduction. Okay, let's go to the next one. When doing a plaque assay, what is the purpose of adding a semi-solid agar overlay on the monolayer of infected cells? A, to stabilize progeny virus particles. B, to ensure that the cells remain susceptible and permissive. C, to act as a pH indicator. D, to keep cells adherent to the plate during incubation. And E, to restrict viral diffusion after lysis of infected cells. Secret answer F, because Prof just loves plaque assays. Right. I'm chuckling because I'm reading the chat. I don't want to distract you with it. But I'm learning about. So gallbladder used an Underwood manual typewriter with manual whiteout tape. And California Norma says manual typewriters burned more calories. And then Les writes, ChatGPT is from OpenAI. Microsoft Copilot is based on ChatGPT. Google has Gemini. Anthropic has Claude and its models. So I'm not familiar with Anthropic at all. Yes, you need a URL and a room name to be brought to the surveys. Okay, we have 20 of 28. Let's see what we did here. Most of you got E, which is correct, to restrict diffusion of the virus after lysis of infected cells. So none of the others are correct. You don't need it to stabilize particles. You don't need it to act as a pH indicator. You don't need it to keep cells adherent. They will stay adherent without an agar overlay. But when virus comes out of the cells, if you had a liquid overlay, it would diffuse everywhere and you wouldn't get a plaque. So that's the reason. Okay, number 3, in particle to PFU ratio, particle can best be described as one of the proteins which makes up the virus, a virus which may or may not be infectious, a virus which is infectious, a virus which is not infectious, elementary, or composite. Patricia writes, RFK's claims about widespread vaccine harm or regulatory fraud on this issue, mainstream scientific consensus does not align with his position. Exactly right. Frankie writes, ChatGPT remains underwhelming. I asked what's the old name of the polio 2C protein. It still gets it wrong. Yeah, but I don't. A lot of other things it doesn't get wrong, right? So I don't know what to say about that. Why it is. I mean, it may be so far away in the literature that it hasn't even consumed it. I just don't know. What's so hard, Costella asked, about looking stuff up? Well, sometimes there's a lot of stuff, and it would take you months or years. And so it's all in these large language models, right? All the information in the world, or a lot of it. And so it helps you. It can be. So that's the thing, to make it where it is useful for humans in their activities. And it's clearly not perfect yet, but it is evolving rapidly. Red Bobby will vote in November. I will vote hard. Also, I put in a lot of phone banking time. Good. That's really great. Hello, Kira. First evening here. We're talking about the new proposed mRNA vaccine for flu. Yes. The FDA says that Moderna has clinical trial results. They want to license it, and the FDA said no. No. Okay. Let's see what we did here. 22 people. What did we get? Yes, it's a virus which may or may not be infectious. So the particle to PFU ratio is simply the number of particles divided by the number of infectious viruses. And that's so particle is either one, both. It's the infectious plus the non-infectious one divided by the infectious ones. And so it's a measure of how many particles made by infection that are infectious. And for some viruses, it's one. Every particle is infectious. For some, it's 10,000. 1 in 10,000 particles are infectious. So it ranges widely over different viruses, and why this is a good question. I mean, there are many things that can go wrong, including not having genomes in the particle, having mutations, and so forth. All right, one more question. If cells are infected at an MOI of 10 in a one-step growth cycle experiment, in the growth curve, you will likely see A, multiple bursts of virus release, B, multiple eclipse periods, C, a single burst of virus release, no burst of virus release, or asynchronous infection. You could give this to ChatGPT and see if it gets it, but I don't want you to do that. I want you to answer it on your own. John got all 4 right. That's good. Frankie writes, a particle is certainly a proton, an electron, or a muon. That's what Einstein said. The world is 5% protons. The universe is 5% protons, 5% neutrons, and 90% muons. Something like that. I love it. John credits the lectures in number 6 this week, which was very good. You know, John, you are listening to them again. I'm very impressed that every year you watch them again. That's how you learn any subject, by going over and over stuff and getting tested and getting more, like you come to office hours, you listen to the podcast. That's how you learn a subject. Now, I know everyone doesn't have time to do all of that, but you know, if you need to learn something, that's what you have to do. And it's impressive for you because this is not your job, right? This is a passion for you. You don't think Einstein said that? Who said it? Somebody else? Let's see. Who said the world is 5% electrons and 90% morons? Oh, Frank Zappa. That's right, Tom. Frank Zappa said the universe consists of 5% protons, 5% neutrons, 5% electrons, and 85% morons. Yes, I'm sorry. Thank you, Tom. It wasn't Einstein. Okay, let's see how we did here. Single burst of virus release. Over half of you got that. So at an MOI of 10, every cell in the plate is infectious, okay? Infected with virus. And so you're going to get a single burst of virus release. Multiple bursts would happen at low MOI, which would be less than 1. Okay, so that's the answer. Okay, folks, you did a good job here on that one. We'll have another one next week. So I'm mirroring the course. Actually, Monday we have the first exam on the first 6 lectures. Let me get back to some comments here. Mark asks, what is mRNA amplification? As in an mRNA being amplified by the cell's RDAP. So, no, there is no cellular amplification of mRNA. So mRNA is produced by DNA-dependent RNA polymerase, right? Pol2 specifically, and that's it. That's the end of the production. If you want to make more mRNA, you go back to the DNA and transcribe it again. So it's not amplified. So yeah, I agree, Quistela. It's good to look things up, and I do that all the time. But sometimes you want a huge field being summarized, or maybe you have a specific question. Like if I'm going out of my field, I can ask it for some tech help, right? So it seems to be pretty good at tech. Like I was asking how to back up a NAS, a network attached storage, right? What's the best way to do it? How to run a VPN, all those kinds of things. For me, that's useful because, yeah, I can go on YouTube and I could look at a video, but there are 50 of them and I don't know which one is right. So sometimes it's good to, but all they do is source what's out there, right? So they're summarizing that and giving a statistical probability of what the next word in the sentence should be. Okay. What are your thoughts on the advantage for an mRNA vaccine? Okay, so the main advantage, it works. It works well. They prevented millions and millions of cases of COVID, right? They got us out of COVID. They could be made very quickly, right? As soon as you have the sequence of the antigen that you want, you can make the DNA and make mRNA and start testing it. So for COVID, it was, well, the 3 trials were done in a year. So if we have a pandemic, we could have a vaccine in a year, or better yet, it would be nice to have them already made, but who knows what the virus is going to be. So I think those are the advantages. What are the disadvantages? The one that I can see is there's a low rate of myocarditis in younger men with mRNA vaccines. But it's usually treatable, and the rate of myocarditis is far higher if you get COVID. So the risk is outweighed by the benefit. And I'm not sure that even applies to all mRNA vaccines. Okay. Vaccination is cheaper than hospitalization. That's why insurance companies want it, right? And Frankie says those chatbots are good at simple programming. Anything I can easily learn in a few days, it can code in 1 to 2 hours with some troubleshooting. But they still allegedly fail at harder tasks. Yes, in vanity, someone will be marketing bootlegged international vacations in the future. Yes. Vincent, if you black out Daniel's background, don't let him wear black because he looks like the computer AI at Red Dwarf. He should be Star Trek's holographic doctor, though. Volunteer him. That would be funny. Yeah, he doesn't usually wear black. He doesn't usually wear black. Hello, Melinda. Welcome. It's almost over, but welcome anyway. Covalently closed circular DNA. That's right. I got a great letter today. Another cardiologist commenting on infectious diseases, saying, BS, you know, a cardiologist is not an expert in infectious diseases. These guys that just are out of their circle. Why are cardiologists anti-vaccine? Not all cardiologists are anti-vaccine. Many of them are pro-vaccine, right? But just a few are outspoken negative and they get a lot of attention. Have I ever met David Suzuki? I have not. I have not. How do they get more than the X, Y, and Z axes? I have no idea. I only know X, Y, and Z. Could a promoter for the late viral genome actually mess up the whole virus replication cycle? It could if it, if it misfired, right? If it wasn't on, if it was off, or conceivably if it came on early, it could also mess up the cycle, yeah. It's all about timing, yeah. Catch the bank robbers by stealing their getaway car. That's very good. I like that. Okay, so for the Hep B recovery is the strong T-cell response triggered by lots of capsid. So I think what happens is giving the capsid assembly modulator gives the immune system a rest because you're knocking down the antigen substantially. The immune system recovers. Then when you withdraw the modulator, you get antigen production, viral antigen, and then the T cells come alive again. That's what they speculate in that paper. Yeah. Okay. The plaque assay princess would know the answer. Yes, the okay, so Nevitz writes, AI is like a primer. It helps you start the search, but you got to do the work to make sense of it and dig deeper, for sure. And Patricia, the best advice in school about learning key concepts, make it your own, understand it the way you get it. You got to go to a place where you understand it and are comfortable with it. And Visto asks, could low functional as an infectious particle ratio serve as chaff to confuse the immune response? I don't think so because they're antigens, right? And they're going to induce an immune response. So they're probably going to cross-react with the other virus. So probably not. I think they're mistakes. Yeah. Yeah, Frank Zappa. Yep. Going to Montana soon. Going to make some dental floss. If I ever get into a situation of having low blood pressure, you could just show me a photo of Vinay Prasad for it would surely skyrocket. No, MOI is multiplicity of infection. How many virus particles you add per cell. So if it's 10, that would be 10 infectious virus particles per cell. Why does your blood sugar go up when you have a virus? Viruses need energy, and a quick way is to metabolize glucose. So maybe that's why they bring it into cells and make lactic acid from it and make a lot of energy along the way. So that may be it. So an MOI of 10 is 10 viruses per cell. Mark writes, asking broad questions of LLMs yields poor results. Providing context is key. Instead of how to back up a NAS, ask, I have XYZ NAS about X amount. Yeah. That's absolutely true. You're right. Robert took a course in virology 40 years ago. I should review my class notes. Carol says, all the cardiologists I work with are pro-vaccine. Yeah, there are very few cardiologists. So there was one during the pandemic, Eric Topol, and then now there's the cardiologist who is heading ACIP, and then there were one or two others, yeah. They don't know anything about vaccines. Just the guy heading ACIP doesn't know anything about vaccines. Molotra doesn't know anything about vaccines. Does Hep B have the same mutation rate as a retrovirus? Yes. It's got reverse transcriptase. Yes. What are we learning about dementia and viruses? We are learning that herpes viruses appear to be majorly associated with dementia. I don't want to say cause yet, but if the more herpes reactivations you have, seems to give you a higher chance of having a dementia. And vaccination against shingles, which is the one herpes virus for which we have a vaccine, seems to make a substantial decrease in various dementia diagnoses. So what you can do at this point is get shingles vaccination. No matter where you are, you're supposed to be 50 or older to get it, but sometimes you can talk doctors into giving it to you younger. I've gotten emails saying that. But get shingles vaccine, get both of them. That will decrease, depending on the study, up to 20% of dementias. Spamlet says axes number depends on symmetry. Quartz crystal has 3 horizontal axes at 60 and a vertical Z. Okay, thank you. Thank you very much. How do you count virus particles? An electron microscope is really the best way to count them. So if you want to do a particle to PFU, you have to count them. Or you have to... You may be able to measure them spectrophotometrically and have a coefficient that converts the absorbance with the number of particles. That's also possible. Yes, like shingles. Well, the shingles vaccine is for preventing shingles, although it's being tested for chickenpox now. But it's the same virus. Yep. What's my favorite virus? Poliovirus, which I spent most of my life thinking about and still do, in fact. Okay. Another example of decoys in biology. Yes, that may be a good topic for a mini lecture. It's a little bit much to do right now, but I'm going to write that down. What happened to myocarditis events brought on by improper vaccination? I don't think that. I mean, experiments showed that in mice, if you put mRNA vaccines intravenously, they caused cardiac damage. But most people still got intramuscular immunizations, and whether hitting an artery or a vein was responsible for myocarditis really has not been resolved. All we know is that there's a low frequency of myocarditis, both with vaccination and with natural infection, right? So natural infection, there's no injection involved. You're getting a substantial frequency of myocarditis. So I don't think that means that it's necessarily the injection. All right, before we go, let me read one poem for you. This is Sylvia Plath, who was an American poet who developed the genre of what's called confessional poetry. And I had 3 poems, but I'm just going to read one. Sylvia had a very tough life. She committed suicide in her 30s, right? And her poems are very powerful. So this one is called Edge. The woman is perfected. Her dead body wears the smile of accomplishment, the illusion of a Greek necessity. Flows in the scrolls of her toga, her bare feet seem to be saying, "We have come so far, it is over." Each dead child coiled, a white serpent, one at each little pitcher of milk now empty. She has folded them back into her body as petals of a rose close when the garden stiffens and odors bleed from the sweet, deep throats of the night flower. The moon is nothing to be sad about, staring from her hood of bone. She's used to this sort of thing. Her blacks crackle and drag. It's Sylvia Plath. I'll read the other two next time. I want to thank everybody for coming, particularly our moderators tonight, Les, Fabi, Tom Steinberg, and Vanity Nutrition. Thank you so much for joining us. Thanks everyone else for coming with your questions. Thank you for participating in the quiz, for having great chat. I really love having you guys here. It's wonderful. We'll be back next week. Let's see. Today is the 11th of February. Next week is the 18th of February, 8 p.m. Eastern. We will be here with more sad news, unfortunately, about the state of science in the U.S., another mini lecture, and some poetry. And, of course, you here with your comments and questions. So have a great week, everybody. Until next week, be safe. Bye-bye.